Buenas tardes a todos. Una disculpa de que Félix no va a poder estar en este seminario, pero para mí es un enorme placer presentarles a Victor Corces. Víctor es una autoridad internacional en el estudio de los mecanismos de regulación de la transcripción génica, principalmente basados en mecanismos de control de la organización tridimensional del genoma y su impacto en la expresión de genes. Víctor es investigador en la Universidad de Emory. Previamente estuvo 25 años en la Universidad de Johns Hopkins en Baltimore, en donde fue director del Departamento de Biología y tuvo muchos otros cargos importantes. Tiene más de 150 artículos publicados, casi 20,000 citas, según Google el día de hoy. Y yo lo conocí en 2014, que estuve en su laboratorio de estancia, y tengo muchísimo cariño, muchísima admiración. Es una persona extraordinaria, muy generoso, y el día de hoy nos va a presentar su más reciente investigación sobre cómo las consecuencias de las decisiones que tomamos en la vida pueden tener en las subsiguientes generaciones. Muchas gracias, Víctor.
Gracias, Rodrigo. Esta es la... ¿Me oís? Esta debe ser la tercera o la cuarta vez que hablo aquí. Y muchos de vosotros no os acordaréis, pero antes era más joven y no necesitaba una silla y subía las escaleras más rápido. Pero como Rodrigo os ha dicho... si no os importa, voy a hablar en inglés porque me resulta muy difícil hablar de ciencia en español. So I'm going to tell you about some recent studies that we've been doing, trying to understand the mechanisms by which the environment affects the germline and causes alterations in the germline that then can be transmitted to subsequent generations and cause phenotypes that you can see in somatic cells. So the system that we use is the following. We take pregnant females and expose them to BPA, which is bisphenol A. Bisphenol A is the stuff that is in every plastic, including plastic bottles. And the plastic bottles that say BPA free, they have BPS and BPF. So there's no such a thing as BP anything free. So we expose the females, and then I'll call the original female is the F0 generation. And then we look at the progeny. The progeny doesn't have any obvious phenotype that at least is not obese. Obesity is the phenotype that we are going to follow. You guys can hear me all the way in the back? Yeah. And either we take the progeny from two different exposures and cross them among themselves, or we can take the F1 progeny, and if it's a male, we can cross it to a control female, or if it's a female, we can cross it to a control male, and then follow what happens. And what happens in subsequent generations is that approximately 90% of the mice are obese. And that goes all the way to the 5th generation. So, given this transmission, then something, since whatever happened, whatever the BPA did, is transmitted from one generation to the next one. Yes?
Sorry to interrupt, but I don't know what BPA stands for.
Bisphenol A.
Bisphenol A. It's just a phenolic.
Yeah, it has a structure. Yeah, I'm sorry. Thank you for reminding me to say that. So it has a structure that is very similar to estrogen.
Okay.
Exactly. So it's a pro-estrogenic compound, and there is a lot of discussion in the field as to what is the actual receptor that is used by bisphenol A. And there's a lot of evidence that estrogen receptor can transmit information by binding to bisphenol A, but other people have information that suggests that there are other nuclear receptors that can also bind BPA.
And naturally, where in the environment the animals can get BPA?
We can get it from everything, so it's in all plastics. So anything that you drink or eat that was in a plastic container has BPA. All the canned goods, all the things that come in a can, in a metal can, the can is lined by a plastic that has a lot of BPA. All the receipts that you get in the stores that are this special type of paper that has tons of BPA. Stuff that you put on your face to look good has so much BPA. Is dental implants that you get at one point in your life, and then you have there forever. They have BPA, and it's leaching continuously, and is continuously in your system. So there's a lot of things that have BPA, but BPA is just one of many proestrogenic compounds that are present, for example, in pesticides. There are things that are present in pesticides that have the same effect, all kinds of things. So we all we are exposed, and if you go and ask somebody to measure the blood or urine levels of BPA in your blood, or anybody here, we all have BPA. And BPA is eliminated very quickly through the urine, like within 24 hours of exposure. However, if you go and measure your BPA levels every day, you always have the same BPA level. So you're being exposed continuously.
And every substance has a concentration where it's toxic and some other concentration where it's not. So what's the observed concentration required to see this phenotype, and what's the observed concentration by natural exposure?
Yes. So obviously we are not all obese here. So we expose these mice to 50 milligrams per kilogram per day. That is a very high dose. We don't have that. We don't have those levels in blood. However, I recently saw that protein extracts that people take to build muscle, they have way more BPA than what we are injecting into these mice. So if you're taking these protein extracts every day, you are exposed to way higher levels than our mice.
How long did you expose that?
So let me go into that. So something has happened to the germline that can be transmitted from one generation to the next, and it can also be transmitted to somatic tissues. Now, this is a F4 generation mouse that comes from a great grandmother that was exposed. Yes.
Just a curiosity. You say that it's transmitted for 5 generations.
Yes.
And I wonder why 5?
Because in the 6 it goes down, and in the 7 is completely gone. But why, we don't know why.
You don't know why.
Yeah, we don't know why 5. So they are fat. All the adiposity is in the abdomen. So if they don't have fat in any other part of the mouse. So if you open them up, this is a control. All this is the extra fat that these guys don't have. And you can see the weight. This is an F3. You see this is the control mice, a whole bunch of them at different ages, and you see the BPA exposed mice are obese. Now, we expose them in a very specific window of development for a very specific number of days. So we are trying to affect the germline, or we are trying to study effects on the germline. So this is mouse development, fertilization. This is the blastocyst stage. This is after gastrulation. That's when the precursor gonadal germ cells the precursor germ cells are going to form. At day 7.5, that's when they form in the epiblast stage of mouse development. And then after that, these cells that are going to form the germline start migrating to where the gonad is going to form. And they arrive there by day 10.5. And at day 13.5, you have differentiation into the male and female sex, and things happen. What happens to the male germline is different from what happens to the female germline. But what is important in this slide is the fact that at day 7.5, more or less, the DNA of the germ cells starts to get demethylated. So this is levels of 5-MC in the germline. So at 7.5, the levels of 5-MC start decreasing, and by 13.5, both the male and the female germline, since they are common up to this point, they are completely demethylated, with the exception of a few transposomal elements, a few AAPs in mouse. And then, if the germline is going to be male, then it starts getting remethylated right away. If it's going to be female, then remethylation happens later on. We can expose the females for just 1 day at day 13.5, and we see the same phenotype. Okay, so we think that the sensitivity of the germline to BPA is due to this window of opportunity where the DNA is getting demethylated. So the model that we have is that BPA either directly or indirectly through causes a change in transcription, causes a change in the binding of transcription factors to the germline. Normally, those transcription factors would not be bound to the germline, and so at this point, when the DNA gets remethylated, the DNA at the binding sites of those transcription factors would become remethylated again. However, if the transcription factors are bound, then they protect against remethylation. So this is based, what I'm telling you, this is our hypothesis, is really based on the results that I'm going to show you, okay? So, and it's based on that because I'm going to tell you the punchline of the final finding is that when we look at the germline of fat mice, we are looking, at this point we are looking at a sperm of the fat mice and the control mice. What we see is the 68 new binding sites for transcription factors. And those transcription factors are CTCF, which is involved in 3D chromatin organization. Zinc finger 143 is another protein that colocalizes with CTCF and is involved in 3D organization. And I think importantly for the phenotype, you can see estrogen receptor alpha and beta, various estrogen-related receptors, androgen receptor, FOXA1, which is necessary to recruit androgen and estrogen receptor to the genome, and PPAR gamma. So these are the changes that we see in the genome. Okay, so the idea then, the hypothesis is that BPA is somehow, probably by binding to a receptor and causing a transcriptional change, is altering the binding of all these factors to the genome, and then that binding is inherited from one generation to the other. Now, this, in principle, this is not something that people would have thought about, because this is transmitted, as I told you, it's transmitted through the male germline. So these changes have to be present in the sperm. So normally people view the sperm as a very specialized cell with a very condensed nucleus, where most of the histones have been replaced by protamines, and protamines form these toroid structures that condense the DNA in the nucleus. Do you guys, anybody here works on sperm? No, just in case I don't want to offend anybody, but this, so this is completely wrong. So the idea that sperm is different, it started different from this. I should say, the DNA has been known for a long time that the DNA in sperm is methylated at specific sites. So DNA methylation could be the source of epigenetic memory from one generation to the other. The problem is, as I told you here, is that in each generation, first of all, right after fertilization, the methylation of the paternal and maternal genomes is erased in large part after fertilization. But then here, again, during the development of the germline, is erased almost completely. Okay, so DNA methylation is by itself is not a good candidate for epigenetic memory in this context for transgenerational memory. Now, Brad Kearns and Sohamud's lab in 2009 published a very nice paper where they show that, contrary to what people had thought, that most of the genome of the sperm was completely covered with protamines, and that a few nucleosomes remain, but maybe that was a mistake, and the nucleosomes were not in the specific places in the genome. They show that the nucleosomes that remain, in their case, they concentrated on looking to see if they were present in genes that were then expressed early in embryonic development. So they show that nucleosomes remain, and they had modifications in H3K27 trimethyl, and that was the focus of the paper. I don't remember if they looked at another crystal modification. And then, since then, several other groups had studied this issue and tried to map the location of nucleosomes in the sperm. And actually, these different groups came with completely different results as to where the nucleosomes are present in the sperm. So we decided to, in the context of this transgenerational paradigm, we decided to look at this very carefully. And we did that using ATAC-seq. So all the data that I'm going to show you it is based on ATAC-seq. And we were able to do that because ATAC-seq didn't work in a sperm, the original method. But this guy here turns out to be one of the best scientists in the world, and he just happens to be my son, came up with a method called OmniATAC. And way before he published it, he gave us the protocol so that we could use it for sperm. So we had, you know, when you have a baby and they cry all the time and you have to change the diapers and you say, why the hell did I do this? Well, sometimes it's a good thing. They give you protocols before anybody else can happen. So the way we look at the ATAC-seq data and we think about ATAC-seq data is the following. So ATAC-seq is a technique that uses TM5 transposes that is loaded with adapters that contain the sequences that you need in order to make libraries to sequence in an Illumina machine. And so the transposase will insert everywhere in the genome as long as the DNA is not covered by something. So it's not able to transpose inside nucleosomal DNA, and it's not able to transpose on sequences that are occupied by a transcription factor. So you end up with little pieces of DNA like this, and you can make a library and sequence them all, and you can separate them by size. The small ones tell you where transcription factors were bound, and the big ones tell you where nucleosomes were present. So you can map the position of nucleosomes and the position of transcription factors. So this is an example of this. So this is a gene in sperm. You can see, so the small reads that tell you where transcription factors are bound, I'm going to call those transposase hypersensitive sites, so THSSs. I'll try not to use that name too many times. But you can see at the promoter of this gene, there is a hypersensitive region where something is bound and is not digested or cut or whatever by the transposase. And then is flanked by two nucleosomes. And then we also did ChIP-seq with a million different histone-modifications, and you can see these two nucleosomes have H3K4 trimethyl. So this is an example. This is where we find where we can map all these transposase hypersensitive sites and nucleosomes. So many of them are in intergenic regions, suggesting they are in enhancers, or in introns, suggesting that they are in enhancers, and at promoters. Okay, so this is telling us something is occupying promoters, and those and maybe enhancers, and they are flanked by specific nucleosomes. So the nucleosomes are that are left in the sperm are present in specific regions of the genome. So this is a genome-wide analysis of the ataxic data. So this is all the promoters in the mouse genome, and you can see that 2/3, more or less, of those promoters in a sperm have a transposase hypersensitive site. That means that something is protecting, something is bound at the promoter, at the TSS, and protecting from transposase insertion. And the most obvious candidate is RNA polymerase and components of the transcription complex. Those TSSs are flanked by nucleosomes, well-positioned nucleosomes, and they have H3K4 trimethyl, K27 acetyl, K9 acetyl. They don't have K9 trimethyl or K27 trimethyl because, so all these histone modifications are typical of genes that are transcribed or in an epigenetically active state. These ones are typical of genes or regions that are not transcribed. And you can see that all the promoters that don't have this hypersensitive region here and don't have position nucleosomes, they have around them over a long distance, this is plus minus 1.5 kb, they have these two histone modifications. And there is H3K4, H3K36 trimethyl, which is characteristic of genes that are being transcribed. And there is no DNA methylation, and there are protamines. This is where the protamine 1 is located. Protamine 1 is located everywhere in the genome of the sperm, but it accumulates at the plus 1 nucleosome. Okay, so what this tells us is that although there's zero transcription in sperm, that a lot of promoters are in an epigenetically active state. They are not transcribing, but it's like they are ready to transcribe. So what are the components of the transcription complex? So I'm going to go very fast through some of these things. We demonstrated using, just by Western analysis, that POL2, phosphorylated serine 5 and serine 2 are present there in the sperm, just the same as in embryonic stem cells. By immunofluorescence, we can detect those things in the sperm. We can do ChIP-seq with RNA polymerase, and this is a sperm, embryonic stem cells, liver. You can see that the profiles are not that different. It's just that in a sperm, the genes are not elongating, so the polymerase is not through the coding region of the gene, as it is, for example, in the stem cells in this case. Not only polymerase, but also I think this is mediator, that is another large complex that is present at promoters and enhancers is also present there. Okay, so this gives us a very different picture of the sperm epigenome. The sperm has promoters occupied by the transcription complex, flanked by nucleosomes with active histone modifications. Many of the promoters are in that stage. And then another subset of promoters are not like that. They have protamines everywhere, and then there are random nucleosomes containing inactive silencing histone modifications. So the sperm epigenome in the mouse is very similar to any other cell. It's just that it's not transcribing, but it is ready to transcribe. So when we think about what BPA can do to the germ cells that then can be present in the sperm or the egg and transmitted from one generation to the other, this is a strong possibility, because by assuming that the sperm is a stupid dead cell, when people think about transgenerational inheritance, they just think about DNA methylation, but then they have a hard time explaining how DNA methylation can be erased twice in each generation and still contain a memory of information. or small RNAs, microRNAs, tRNAs. And so what I'm trying to tell you is that there is way more things in there that can be affected by the environment, not just BPA in our case, but by many things in the environment. Now, this is not just mice. So we've done, so these are, a lot of, we can map a lot of transcription factors in these distal enhancers that we can map with a TAC-seq, we can map the presence of something. Then we can use the data to do a footprint analysis, and we can try to guess what transcription factor is present there. So you can see FOXA1, FOXA2, all kinds of FOX proteins, estrogen receptors, different nuclear receptors, CTCF, and different other proteins. So a lot of different transcription factors are present in the sperm. And it's not only in the mouse, but if we do the same type of analysis in humans or in monkeys, and we look at regions of the genome that are conserved, they are present in the same location in the genome of these three mammalian species. So it's true also for humans. Now, what kind of transcription factors are present in the sperm? The ATAC-seq doesn't allow, it only allows us to do a footprint analysis and guess what transcription factor is present and look at based on the nature of the motif. And so we did Western analysis for a whole bunch of them. It doesn't really matter. Then we confirmed that by immunofluorescence analysis, and we show that really the proteins are present in the sperm. And then we did ChIP-seq, for example, in this case is FOXA1, estrogen receptor, androgen receptor. You can see this is an enhancer inside this intron of this gene. You can see they are all present there. H2AZ is a histone variant that we think is very important in the memory mechanism, and is in the two nucleosomes flanking the binding sites of these transcription factors in the sperm enhancers, is also present there on H3.3. Okay, so all these proteins are present in the sperm genome, and so we are defining enhancers based on the fact that these transcription factors are present bound to the genome in distal regions far away from promoters. We can define enhancers in a more classical way in the chromatin field by looking for regions that have H3K4 monomethyl and don't have H3K4 trimethyl, and they have either H3K27 acetyl or K27 trimethyl. So you can see this is a collection of enhancers defined this way in the sperm epigenome, and they are in an active state, and some of them are in a bivalent state. But it's like they are ready to go. They are not going anywhere because there's no transcription, but they are ready to go. And if we look at these enhancers and what happens later on during embryonic development, Bing Ren's lab has characterized enhancers in different tissues of the embryo when they already have bones and liver and brain and stuff like that. And these enhancers are going to be active later on during embryonic development. And we can define super enhancers the same way. Okay. And more importantly, for our own interest, CTCF is present in the sperm. So, as I said, CTCF, and you know from the work of Myra and Felix, that CTCF is very important in folding the chromatin inside of the nucleus. And until now, we've always thought that the sperm chromatin was somehow packed there like crazy, not in a normal way. But CTCF and cohesin, so this is sperm CTCF, sperm cohesin, this is round spermatids, which are the stage before the sperm matures. These are embryonic stem cells. You can see they are present in many common sites in all these cells, although some of them are specific for sperm. And this just gives you a genome-wide view. And for example, just to show you, these are all the sites occupied by CTCF in a sperm, all the sites occupied by cohesin in a sperm. This is embryonic stem cells. So the sperm, the embryonic stem cells have additional sites, but the sperm has a very large fraction of the sites present in other cell types. And if we do high C to look at the folding of the chromatin, we see that CTCF makes loops. These dots here are loop, the signal corresponds to loops made by CTCF, and these are very similar to the ones that you would observe in any other cell type. So this is sperm, embryonic stem cells. This is an lymphocyte type cell line. You can see the domains that you see in a sperm are very similar to stem cells, not so similar to other differentiated cell lines. Okay, so the genome is folded in the same way. So I'm going through all this and I'm telling you about this because if you go back to the sites that I told you were transmitted, the new 68 sites that were transmitted in the sperm from one generation to the other, the proteins that were involved were CTCF, FOXA1, estrogen receptor alpha. So that's why I'm making the point that these proteins are in the sperm. Now, in the oocyte, it's harder to get enough oocytes to do ChIP-seq, for example, with CTCF. But we have done ATAC-seq in oocytes, okay? And we have looked at immature oocytes that are arrested in the GD stage, which is the prophase before meiosis 1. And then the mature oocyte that is arrested in mitosis, in the metaphase, I should say, no, in the metaphase of the second meiotic division. This is what the sperm encounters. So Rodrigo didn't tell you this, but I'm a chemist. So I don't know any biology, really. I have an undergraduate degree in chemistry and a PhD in chemistry. So all the biology that I know, I learn it as I go. So I never thought about, what is the stage of the oocyte when it gets fertilized. But it turns out that it's in metaphase. I mean, not metaphase. Yeah, metaphase. So that is like the chromosomes are all condensed and a lot of the stuff maybe has come out of the chromosomes. And so if you are thinking about transgenerational inheritance that has to be maintained in the chromosomes, what is going on in this mature oocyte? So we did ATAC-seq in all these things. And I will tell you that in GB oocytes, when we look at in this slide we were looking at all the TSSs. So we can find hypersensitive sites to the transposase at the TSSs of the same promoters that contain the transcription complex in a sperm. And I will show you the data for that in a second. If we look at distal sites, presumed to be enhancers. These are all the enhancers that contain transcription factors in the GB oocyte. These are the ones in the M2 oocyte. So going through metaphase, it really didn't do anything to the TSSs of most genes in the oocyte. We lost a few, but we gained a whole bunch of them. And you can see they are flanked by perfectly good nucleosomes. Okay? And we can do the same thing with CTCF. Okay? So, based on ATAC-seq analysis in the oocyte, we can see that there are promoters that must contain the transcription complex. There are enhancers that must contain transcription factors bound, and we can guess which ones they are by looking at the sequence at the summit of the peak of the ATAC-seq data, and there are CTCF sites. Now, this tells us that the epigenome of the germline is very rich in things that can be altered by the environment. But can that epigenome be transmitted to the germline? Can that information be transmitted to the embryo, after fertilization? So this shows you all the TSSs, all the promoters of all the genes in the mouse genome in a sperm. I showed you this before. This is RNA polymerase. So this is the ataxic signal. This is RNA polymerase from ChIP-seq. So you see that there's a very good correlation. So then I take the ataxic signal to infer that there must be RNA polymerase and other proteins present in cells where we don't have enough cells to do ChIP-seq and really show that those proteins are there. But in the GBO side, you see it's the same promoters that are active in a sperm, not active, but occupied, that they are occupied in GBO sites. In the M2O side, this is DNA-seq that we got from these guys here. This is very early zygote. This is a later state of the zygote, so this is the one-cell zygote. So it's maintained in the paternal. This is the paternal chromosome. This is the maternal chromosome. Paternal, maternal. And this is the 2-cell stage, the 4-cell stage, the 8-cell stage, the modular stage. So the same promoters that were occupied in the germline, in the gametes, are still occupied in the embryo. So this suggests that maybe the reason that whatever information is in the gametes is going to be used then early in development and is going to be important for early embryonic development. So when we think about transgenerational inheritance and affecting the epigenome of the germline, we can guess that maybe it's going to affect also the development of the embryo. Now, this is only one example of this, but the other possibility for transmission of epigenetic information is histomodifications. So this shows you just one example, and I have a slide like this that I don't have here, but I'm analyzing it genome-wide. But this is true genome-wide. This is H3K4 trimethyl and H3K27 trimethyl. At this specific gene, in the promoter, this specific gene in a sperm, in the oocyte. You can see that the paternal chromosome has lost this histomodification, and it wasn't there in the maternal chromosome, is not present then until the late 2-cell stage. That's when transcription starts again. So in my view of epigenetics, transcription factors, I don't know if you, I guess I have been talking about this in sort of in a subtle way, but transcription factors are at the top of the cascade that initiates transcription. And histomodifications are a consequence of that initial event that is the binding of a transcription factor to an enhancer. Everything else, all the histomodifications, DNA methylation through the coding region of the gene is a post-initiation event. And from my perspective, it cannot carry epigenetic information. I know that this is heresy for many people in the field. And so that's why I'm emphasizing transcription factors so much. Okay?
I'm sorry. This is a short question perhaps. But I'm not neither a biologist. My understanding of the first stages of development is that these cells are pluripotent or something like that.
Yeah.
So why do the sperm doesn't have the imprinting of transcription factors like embryonic stem cells? Because that should reflect the pluripotential that these cells have, right?
Well, it's very similar, no? So until now, I only show you promoters, but the sperm is, in terms of the transcription start site, in terms of the promoter, is exactly the same as...
In this one, but in the previous ones, you were showing that the embryonic stem cells were more protected in some areas than the sperm cells, right?
Oh, you mean the embryonic stem cells in... Yeah, but those are embryonic stem cells in culture, and they are, in the way we culture those cells, they are in the epiblast stage. So by that stage, they are not pluripotent anymore. Yeah. So the cells,
So the pluripotency is not equivalent of having every single promoter ready to be transcribed.
well, it's just that some promoters have changed by that time.
But even in your last slide, not every single promoter.
Exactly. So this promoter, if you look at these promoters here, they are all involved in immune response, and it's all very specific functions, very late in very differentiated cells. So at this point, they are all silenced. They don't have the polymerase there. Okay, so you're gonna so this also addresses your question. You're gonna tell me this is different, and you're right. So these are all the enhancers occupied by transcription factors in the sperm. This in the sperm, in the oocyte, so it's different. And then in early embryonic development, data from Weiche's lab with Ataxic, the data is not very good, but you can see that they are not exactly the same as the sperm. And what they are, if you follow them, is whatever is common to the sperm and the oocyte is what is transmitted to the embryo in terms of enhancers. And a similar thing with CTCF sites. These are all the CTCF sites. So I'm going to skip this because it's getting late. So first I told you there are transcription factors and the transcription complex in a sperm and the oocytes. Now I'm trying to show you that that information is transmitted to the embryo, at least part of it, and that what we tend to see is that what is transmitted to the embryo is what is common to the oocyte and the sperm. Now, I also told you that in the sperm, the genome is folded by CTCF and other forces in the same way as in normal cells. So part of the role of CTCF is controlling what enhancers are going to interact with what promoters. So what we think is that in the sperm, interactions between enhancers and promoters are already pre-established. That's what the high C in a sperm tells us. I didn't show you the data, but that's what it tells us. So, for example, if we take, yes?
Sorry, out of curiosity, if the pattern is the same, the size of the nuclei of sperms is exactly the same size as the normal cells?
No.
It's smaller.
Is way smaller. So the volume of the sperm nucleus is only three times less than a lymphocyte. Lymphocytes have very small nuclei, but way smaller than many other somatic cells. But in the nucleus of a somatic cell, there's a lot of empty space. So you can, it's like an accordion. You can still fold the accordion so that it's folded the same way. It's just occupying a smaller space. So here I'm showing you a meta-analysis of all the interactions of all the enhancers that have FOXA1 as androgen receptor and estrogen receptor with the target promoters. So I should have another slide here to explain this. But essentially, what we are looking at is, at the same time, all the interactions between enhancers with FOXA1 and promoters of genes in a sperm. And this dot here represents all the interactions. And then we are looking at different times during embryonic development. And this is high C data, so the sperm high C data is ours, and I showed it to you. The embryonic data is from Weiche's lab, and it was done using very few cells. So the data, the quality is just not there. But you can see that when we look at the same enhancer promoter interactions early in the zygote and later in the zygote, and the early to cell, late to cell, 8 cell, ICM is the same. These interactions are pre-established in the sperm genome. And we can do the same thing in the oocyte with Hi-C data from these guys, and you can see that interactions in the oocyte, which are different from the ones in the sperm, are also pre-established. Now, to make things more confusing, and I don't know if it's worth going through this, but if you think about it as a geneticist, the first slide that I showed you with the crosses between the mice, if the germline of the male is altered and then you cross it to a control female, how is it possible that 90% of the progeny have the obesity phenotype? And then if you repeat that cross, how can it be transmitted like this? Because if you just follow the genetics, it doesn't make any sense. And that is because when you look at interactions, and here we are looking at interactions between CTCF sites, in the sperm, they are present in the paternal genome during development, but they are not present in the oocyte, and they are not present in the maternal genome early on, and they only appear later in development. And the same thing is true for oocyte CTCF loops that are present in the maternal genome early in development, not in the paternal genome, but appear again. So what this is telling us is that during early development, the folding of the genome is specific for the maternal and the paternal chromosomes, but at one point, probably around the 4-cell stage, the two of them become the same. Yes.
Seems to be pretty much shared.
Right. I mean, I can't really make very strong comments about this because the quality of the data is just not there, but that's what it appears. Some of them are, and some of them aren't. Yeah. Oh, I'm going to show you that in a minute. Okay, so we go back to here. Now we go back to the fat mice. So these are the changes. So in the context of everything that I told you, now this is what we find in the sperm of the fat mice, new binding sites for all these proteins. So what does that mean? So these are examples just to show you that the data is, this data is actually very robust. So these are two replicates of ataxic in the sperm of F1, F2, F3, and control. And this is a new site that we see. So this is a site that doesn't change, or this is a site that doesn't change. This is a site that changes. It's not present in the control and is present and maintained and from one generation to the next one. And this is an ataxic peak. But we can go and look at the sequence and we see that it's a CTCF motif. So we have not demonstrated a CTCF, that is CTCF, but there's a motif for CTCF there. So we are assuming that it's a CTCF site. These are other sites. This is an androgen receptor site. This is a SOX5 site. So we have 68 of them, okay? Now, we have done BSEq in all these generations of the sperm and BSEq at a single base resolution so that we can identify whether there are changes in the methylation of the sites of specific CPGs in the motif of the protein. And this tells you this analysis. And you can see, I don't know if you can see it, but this is a CTCF site that changes, okay? So it's not in the control, it's present everywhere. When we look at the DNA methylation at this specific site, This site is hypomethylated in the fat mice. So this comes back to my original hypothesis that, in this case, CTCF is binding, for whatever reason, BPA caused CTCF to go and bind to a place where normally is not present at the time when the DNA was demethylated. That site normally is methylated. And then in subsequent generations, the site stays demethylated because CTCF is protecting from remethylation. So this is what I just told you, okay? Okay, so I don't know if this addresses some of Rodrigo's questions, but I'm telling you about 68 new transcription factor binding sites because they are very robust. There's no signal in the control, and there is a lot of signal in the fat mice sperm. But there are other sites that change. It's just that they may go from a level of signal of 1 to a level of signal of 3, you know. But and so we haven't studied those. Those are less robust. But what we can see when we do high C in the BPA sperm, what we can see is that those new CTCF sites are making loops that are not present in the control. So here is a case, and here you have a meta-analysis of sites with different standard deviation from the control. You can see that the signal of CTCF in the BPA sperm is much higher by 1 standard deviation or by 2 standard deviation. This is a specific site. Okay, so these new CTCF sites are making new loops. And what are those loops doing? So we see an obesity phenotype. That means that something is happening in something that we don't really understand. Something could be happening in the fat tissue that is causing the phenotype. Something could be happening in the liver that is changing metabolism and is causing the phenotype. Something could be happening in many other tissues that are causing this. So we've gone and done the same analysis in the fat tissue of these mice. And we find a whole bunch of new sites that are present in BPA but are not present in control. And in this case, we find also a lot of sites that are present in the control and not BPA. So there are sites that are gained and sites that are lost. And we find similar proteins: androgen receptor, estrogen receptor alpha, PPAR gamma. All these 3 transcription factors are present in this site, or CTCF in this site. This is now the fat tissue. And there is a huge change in transcription in the fat tissue. Now, I'm almost done. Stay with me one more minute. So, if you think about what is happening, what I told you is that there are new sites and they are reproducibly present in the germline. That explains the transmission, but it doesn't explain the phenotype. Now, in terms of the phenotype, it's possible that the same sites that are present there in the germline and presumably in early embryonic development, then they are maintained during cell differentiation so that they are present in different tissues of the adult. We don't see that, except for one case, which is the one that I'm going to tell you. The other possibility is that these new sites that are in a sperm, they are present there early in development. If you remember, the germline separates from the somatic cells at the epiblast stage. And so up to that point, they are maintained, and the germline maintains those changes. But then in somatic tissues, as they differentiate, those initial changes cause a second wave of changes, and maybe a third wave of changes. And those are the ones that are present in somatic tissues and are responsible for the phenotype. I don't know if I explained that well. So you don't necessarily expect the same changes that we see in the sperm to also be present in the adult and be responsible for the phenotype. Now, there is one case. So I forgot to tell you, if we look, for example, at all these changes in transcription factor binding in the fat tissue, many of them are associated with genes involved in obesity based on GWAS studies in humans. So probably the obese phenotype is very complex, and it arises by the expression of many genes, and we don't have causality in the relationship between these changes. There is one gene that explains everything. So that is this new site. You can see it's not in the control. It's present up to the F5 generation. So here's a mouse. In this replica, in the first generation, this was probably one of the 10% mice that was skinny. But it's present in the sperm of the fat mice. This site in the sperm of the fat mice has H3K27 acetyl. And it's not a promoter, it's an enhancer. Okay, so it's marked as an active enhancer in the sperm of the fat mice. And it has H2AZ, which I didn't go into details, but we think is very important in the memory. It's not just DNA methylation, but we think that H2AZ is important in that. Now, someone named Suzanne Mantrup, who studies adipocyte differentiation, has characterized all the enhancers that are important for adipocyte commitment and differentiation. And this site here was characterized by her as an adipocyte-specific enhancer. And it binds, based on the ataxic, binds CTCF and another transcription factor called NKX2.2. Okay? Now, the COBL gene stands for cordon bleu, and it's a stupid gene that encodes a protein that interacts with actin. So there's not an easy way of explaining how interacting with actin is going to make you obese. But doing high C, we have okay, so this is still a sperm. I'm going back to sperm. Analyzing the high C data in the sperm for the fat mice and the control mice, we see that this specific enhancer interacts with several genes in this region here. So these are twofold higher significant interactions. This is just for Maya and Rodrigo in BPA minus sperm. And one of these genes that I have never heard of is DSTM2A, and is a gene involved in obesity in humans. It's a gene that turns on PPAR gamma, which is the main transcriptional activation of all the adipocyte differentiation process. So you can see you can see the difference in interactions between control and BPA here. Okay, so we think that this gene could be a real candidate for the phenotype, and it would be a transcription factor that then initiates a cascade of events in the fat tissue of the fat mice. Now, what we like about this gene is that not only is it present, okay, so also this is not a gene. This enhancer is transcribed, and it makes an eRNA in the fat mice, but not the control mice. But what we like about it is that the alteration in transcription factor binding is also present in the liver and is also present in blood. So that if everything is true that I told you, which I don't know if I could convince you or not, but I believe it because for me it's religion, but we could use this as a diagnostic. So if people are obese because they were exposed to endocrine disrupting chemicals like BPA, they should have a signature of this enhancer in their blood. So this is the one thing that we would like to do. So I'm finished here. Let me repeat just a take-home message for you guys to take home and tell your kids. So the germline has a very complex epigenome. It seems, based on these effects of the BPA, that the environment, a lot of stuff in the environment can affect the epigenome of the germline. And the epigenome of the germline is in part transmitted to the embryo. So it makes sense that these alterations can then be transmitted to the embryo. And then they can cause effects in somatic tissues of the adult. Now, most of this work, a lot of this work, was done by Yoon-Hee Jang, who's an amazing postdoc in my lab. And recently, a few people have helped her a little bit, but she deserves all the credit. So thank you very much, and I hope I made sense. No,
It's a naive question first. These obese mice that you're showing with the controls are eating the same thing as the controls, or are eating more? I mean, I want the relation with the behavior. Is these obese mice
behavior?
are eating more
Oh,
than
yeah,
the
yeah.
control?
No, they are not
They
eating
are
more.
eating the same.
Exactly the same, yeah.
And they are obese. Okay.
Yes.
So it's not the behavior.
No.
Now, the second point, all this relation with these other genes, are there some genes that are related with cancer?
So we haven't analyzed that, but that's a good point. So BPA, other people have described a high incidence of testicular cancer, a high incidence of breast cancer. We know that our mice, by the time they are a year old, if we keep them that long, they all get tumors in different places and they all die from tumors. So, and other people have published that exposure to BPA causes behavioral phenotypes, also causes phenotypes. We observe very frequently in our mice that they will spend day and night going like this in circles. I don't see when they have time to eat, but they are still fat in spite of the exercise. So your point is very well taken. Exactly. So we think that a lot of these changes that we see are not related to obesity. They are related to these other phenotypes that we don't analyze but other people did. Yeah.
Two questions. I was wondering about it's very interesting that you see the CTCF groups specific of each germ cell that then adapt for the other one and then the one is because have you thought about a possible mechanism for that to happen? Is it possible that during cell division that the two alleles are together, then I don't know, have you thought about that?
So the only mechanism that I can think of is something that Peter Fraser has found. And he's talking about it. So I guess I can talk about it. So he has done a lot of single-cell high-C during early embryonic development. And what he sees is that at the 4-cell stage, the two homologous chromosomes pair. So I don't know if you think that this is hand-waving, but because the homologous chromosomes are pairing, and there are a lot of pairing phenomena in plants, for example, where pairing between homologs allows the transmission of DNA methylation patterns between homologs. So the way we explain it is that at that time of development, when the homologs pair, then they exchange some information that causes the CTCF loops to be the same in the two chromosomes. I don't know why that would be, but
there are some evidence, all papers from Schindler's lab that link actin dynamics to shuttling of nuclear receptors into the nucleus. That's in liver, controlling lipid metabolism.
yeah, maybe. But we don't see a change in transcription of that gene, and we see a change in transcription of the
So
other
the
gene.
transcription is not high.
For that gene, no. Yeah. Yes.
So usually the nice thing about biology is that it's never black and white, no? But what you're showing us here is a phenomenon that is black and white. So you have genes or transcription factor binding sites that are in one condition and not in the other. I'm curious to know if you have sequenced the 10% of animals who were exposed to BPA that didn't develop obesity, if they have exactly the same pattern that the other 90% of animals who are obese.
No.
Because that will solve my puzzle.
Yes, I agree with
You
you.
have a perfect pattern, and that's rare.
I agree with you. We always throw away those mice. Now, what we are doing, we are in the process of doing is that I mentioned that in the 6th generation, half of the mice are not obese. And then what we've done is cross the non-obese mice among themselves and the obese mice among themselves. And then in the 7th generation, none of them are obese. So we are doing a taxi in all those mice, which I think addresses the same question. But yes, we should have been looking at that 10% and we didn't. And unfortunately, after you do all this, then and you throw them away, then it's hard to go back. But
Naive and probably silly question. Epigenetics is very dynamic. How come a single administration of the drug can be transmitted through several generations if it is epigenetically based? Are you positive that there is no change in genes? Because this way of transmission, well, it doesn't fit either, because 90% to 10% doesn't fit.
you mean that there are no mutations?
That's right.
Yeah. So we, I mean, in theory, we could look at the genome wide to see if we look for mutations, and we haven't done that. What we have done is look specifically at these new sites, and there we don't see any mutations. Yeah.
Surprising.
I think there are several. I mean, it's surprising, and then it's also surprising that after the 5th generation, the whole thing just falls apart. What is special about that? And I think there are several reports of this type of transgenerational effects that after the 5th generation, they go away. And I don't know what is special about that, but.
Have you covered your epiphenotypes profiles with obese mice?
Yes, so we have, and we don't see any of those changes. Right. So we were just looking at mice, at the sperm of mice that were obese for different reasons. We didn't look at a fat diet transgenerational effect. It's just fat, the sperm of fat mice. We didn't, yeah.
Have you observed any reduced fertility in the BPA mice or in the progeny?
We haven't, but we haven't studied it carefully. But people have reported this, that they make fewer sperm and they have smaller testes. People have reported this type of stuff, but we just don't pay attention. We just cross a whole bunch of them and they have babies and we are happy.
So, what do you think is driving the changes in methylation that will allow the one to that's the first one. And then you said that your mice eventually develop tumors, right? Which is something that you observe whenever you have a deficiency for CTCF. So that question is, you observe here a gain of CTCF binding sites. Do you observe the loss of CTCF binding sites?
Okay, so let me go to the first one. So Our hypothesis is that CTCF binds to new sites at the time when the DNA was completely demethylated. And once it binds, then it prevents remethylation. Now, why is it binding only to those sites? There are thousands, I mean hundreds of thousands of potential sites in the genome. So when we saw your paper on CTCF and RNA and affecting specifically only a subset of CTCF sites, I wonder if it's something like that. You know, why is CTCF only binding to these new sites instead of just every possible site, right? If my hypothesis was true, it would bind everywhere, not just the specific sites. And we don't know, we haven't looked to see if these specific sites, are they close to binding sites for estrogen receptor? I don't know, we haven't.
Because they can interact, the estrogen receptor can bind to regions that are also by CF. So one possibility could be that if you are treating them with physical A, then you are activating the estrogen receptor and it binds to the CF.
Right. And your other question, I think I didn't have an answer for it, but I don't remember what it was.
If you are losing CTCF binding sites.
No, we are losing some sites when you just go through the motions and do all the statistics and do all the things. It looks like there are some CTCF sites that are lost, but when you actually go and look at it, it was a very small little peak before, and now it's gone. But so we think that we are not losing anything actually. Yeah.
Yes. My question is about what Dr. Rio said, that nothing is black and white, right? So I can remember a working where they analyzed methylation patterns in AAV that are these retroviral sequences, and one example of transgenerational inheritance in mammals is the agouti mice, right? In which mice that is agouti color can have, in the F1 you can have mice with any coloration pattern, right? And despite of the work, I think the discussion of of this example is that the most of the epigenome is actually getting lost in the generation, right? I think that is my question is, how do you think that you can compare these two phenomena, right? Your example is pretty regular, you can see group generations and the sites are conserved and so on. But there are other examples that are not as...
You mean like in the agouti mice? You see mice, so you see yellow mice and brown mice, but then you see mice with the spots.
Right.
Is that what you mean? I mean, it could just be the phenotype. So the spots just tell you that there is a stochasticity there, and that some cells are being turned on and some cells are being turned off. In our case, it's not a visible. I mean, the phenotype is either adipocytes differentiate and make way more pre-adipocytes differentiate and make way more adipocytes, or they don't. So you could have some of the original cells, and some of them are doing the same as in the agouti mice and becoming yellow by making adipocytes, and some of them may not. But in the end, we are just looking at a fat mouse. So we can't distinguish. We don't have the possibility of distinguish this stochasticity in the, yeah. So in that sense, the skin phenotype, the coat phenotype is much more sensitive to look at this, to look at position effect variegation phenotypes. But we can distinguish that in the fat. Oh, she had a question there. Hold on a second.
I don't recall the question, but you showed the pattern of a black seed cancer in sperm oocyte, and you compare it with the early embryos. And it caught my attention that the pattern is completely classic cycle and cancer in embryos is overlapping
So I didn't show you that, but we only know this in the sperm. But what it looks like is that the ones from the sperm that are transmitted to the embryo, they all have H2AZ. So those enhancers have H2AZ in them. The ones that are not transmitted, they don't have H2AZ. But so that would mean that they know if they should be transmitted or not. We don't know in the oocyte because we can't get enough oocytes to look at H2AZ. Thank you.